So we'll go on to our next speaker now, who is Sam Thomas. Hopefully he's here and ready to museum in London. And he's the Urban Nature Project Biodiversity Officer. And he's going to be talking about that project, the Urban Nature Project, and which is quite a major new project with the museum and other partner organisations, and how we might be able to get involved in it. So hopefully I can hand over to Sam, who I've lost on my screen at the moment.
Yeah, I'm here.
Brilliant, there you are. Perfect, thanks, over to you.
Cheers, I'll just try and share my screen. Right, is that working?
Yeah, that's working, thanks.
Perfect, thank you. Yeah, so I'm Sam, I'm the Biodiversity Officer for the Urban Nature Project at the museum. And this is a major new nationwide collaborative project led by the museum with a couple of different elements. So there's a big program of different scientific, educational activities around urban nature across the UK, as well as a redesign of parts of our garden at the Natural History Museum. So I'll be mainly talking about the scientific side of the project today. So as we've heard, and as we're all very aware, the state of nature in the UK is not ideal. Nature is very much in trouble, and that applies equally to urban nature as nature in more rural places. With increasing urbanization, increasing density within urban centres, and the impacts of climate change likely to be felt particularly severely in towns and cities, it's really important, never been more important, to appreciate, understand, conserve nature in our towns and cities, and appreciate the role it plays in making them livable places. So the vision of the project is to start an urban nature movement, and that has a number of different elements: creating new opportunities for young people to interact with urban nature across the UK, developing scientific tools and skills to better understand and protect urban nature, maximize its benefits for society, and transforming parts of the museum garden in a sensitive way to create a really amazing example of urban wildlife, a space where we can do research, test methods for management and conservation, and really use the gardens at the museum to raise awareness about urban nature more widely. So I'll talk a bit more about the second two of those, the science program within the Urban Nature Project, which is the area I work on and is likely to be the area of most interest to yourselves. So what we're doing here is we really want to support evidence-led nature conservation by working with as wide a range of different UK biodiversity organisations who have an interest in urban biodiversity. And we want to create toolkits, methods, etc., that can improve monitoring and management for urban habitats within the UK. And we want to help develop skills for urban nature monitoring and management, which will include new urban nature training content through the UK Natural History Training Programme run by Steph Holt at the museum. And we want to develop new methods of monitoring urban nature and generate data that we can share widely. So we've got an amazing series of data from the museum gardens, running back for the roughly 20 years it's been in existence. And we want to continue to build on that with traditional monitoring, the moth trapping, the vegetation recording, etc., the ad hoc wildlife recording. But we also want to bring in new methodologies, such as DNA-based monitoring and acoustic monitoring, that will allow us to improve our understanding of the gardens at the museum and partner sites as well. And we want to make sure that as we develop these techniques for urban biodiversity monitoring, that they're scalable, that they apply across sites across the whole of the UK and are usable by others, not just ourselves, and that they look at both the biotic, the biodiversity side, and the abiotic, the environmental, etc. side. So looking at a whole community monitoring approach. And we want to use these methodologies to study the benefits to biodiversity of habitat restoration and creation as we are working on the museum gardens. We can develop a better understanding of what works and what doesn't using these methodologies, and share that knowledge going forward, particularly focusing on the really data-rich, well-recorded area that is our gardens as a in-depth case study of what you can find out about an urban site. So to go into a few of those methodologies that we've been experimenting with, firstly, metabarcoding, DNA metabarcoding, and this is looking at the traces, the fingerprints of DNA, if you like, that are left behind as organisms interact with their environment. And it's taking mixed samples of DNA, so that could be soil or a Malaise trap sample, extracting, sequencing all of that DNA at once so we can generate rapid, cheap, repeatable lists of taxa or species for samples in a way that would not have been previously possible. So this was all kicked off by a need to really understand the gardens at the museum and how the changes that we're making influence what's present, to track our successes and failures and make sure that we manage the gardens as well as we can going forward. But it's evolved into a wider pilot study of how metabarcoding methods can support practical nature conservation, both in urban spaces and more widely. We're focusing on invertebrates, and we're trying to take as wide a range of samples from the habitats within our gardens as possible. So that's soil, water from the pond, pitfall traps, Malaise traps. And we're also sampling at a number of national partner sites, which are currently other museums through the UK. So what we found initially is that sampling from soils worked pretty well. Pond water sampling mostly worked very well, but we had some issues with Malaise trap samples and extracting the DNA from those samples in a way that we could retain the invertebrates in the sample in a useful state, whilst also getting a representative sequence data for what we could see was in the sample. So during the last couple of years of the project, the experimental phase, we've managed to up that for our samples from around 50% recovery rate to around representing 90% of things turning up in our sequences that are found in a visual sort of a Malaise trap bottle. So that's really exciting and shows the potential for this to actually enable rapid representative sorting of those Malaise trap bottles that have previously taken such a long time and have potentially languished on people's shelves for that reason. So the other side of it, the soil sampling, we've had some really interesting initial results from that, and we're still in the sort of data analysis stage of this at the moment. But we're seeing huge levels of diversity and differentiation in the soil organisms, even between really closely adjacent sampling points in our small urban garden in central London. So that's all going, it's all really exciting stuff, and we'll have proper results for that soon. Of course, with any DNA barcoding based project, you're generally only as good as the library, the reference library you have access to, and that's the sequences, the pre-existing named sequences that we compare the sequence data that we generate against in order to generate species or taxon IDs for each of our sequences. So as part of the UMP, we're working with projects within the museum, other projects such as Brilliant Butterflies, Darwin Tree of Life, and more widely projects like Bioscan, to really rapidly move forward the availability of reference barcodes, so we can actually make this methodology work as rapidly as possible in terms of being able to assign names to sequences we generate that are based on reliable reference sequences from the UK, preferably. The other side of the methodology development that we're doing is acoustic and environmental monitoring. So the traditional biological recording and eDNA monitoring give us really good taxonomic snapshots, species lists from a time and a place. But acoustic monitoring has the potential to give us much more fine level detailed data across time with repeated regular sampling automated. So we want to monitor levels of natural sound, human sound, and a range of environmental variables from the museum gardens. We're installing a permanent wired network of sensor points in the new gardens, with 25 initially going live in 2023. Each of these will be a modular Raspberry Pi based device with a terrestrial microphone, some of which will be ultrasound capable for bats, etc. And some of which will also have microphones in other understudied environments, so hydrophones in water, contact microphones in soil, deadwood, etc. And each of which will have a range of environmental sensors, looking at temperature, humidity, pollution, things like that. So what we're expecting to get from this is not taxonomic data. We're not aiming to be able to identify species from our acoustic recording, but we'll be analyzing the recordings using acoustic indices. And what these provide is a way of comparing different types of sound. So bioacoustic sound, that generated by nature, versus human generated sound, and maybe a level down looking at the levels of the sounds of insect flight, invertebrate activity, or bird song across time. And this network has huge potential because nobody has really installed a dense sensor network in a place like this, particularly not with one with such a wealth of other biological data available. The sensors are modular, so we can change the different devices are modular, so we can change the different sensors that we have in response to the interests of different researchers. And this network will be available for NHM and external researchers to come and use if they're interested in that. So that's the sort of methodology development, and then there's bringing all that together in a way that we can share the work we're doing, learn from other people in the sector, and get the results out there in a way that benefits people. So we want to bring together as many urban nature practitioners as possible and talk about what priorities are for the sector and how we as the Urban Nature Project can best contribute to helping with those. And the main way we're doing that is through a series of ongoing workshops. We've had 2 so far, one around what we should monitor in urban areas, and one around the challenges of managing in small urban spaces. Those were run in collaboration with the London Wildlife Trust. At those workshops, the potential for an ongoing urban nature knowledge sharing network was discussed. There was quite a lot of interest in this, but obviously there's still a lot of questions around how that might work, be resourced, whether we're the best organisation to take that forward, or whether there are pre-existing organisations that might be better placed, but it's something we're interested in exploring. We want to make sure that when we're doing things within the gardens, whether that's creating new urban grassland, depaving an area of paved courtyard to create some brownfield habitats, or relining and replacing our pond, that we use the methodologies we've developed to look at that before, during and after the works, so we can learn what worked, what didn't, and share that knowledge in a series of case studies, focusing on each of those elements. We want to bring together parts of this into a digital toolkit or series of toolkits. The potential topic for which could be around what we should be monitoring in urban environments, quite broadly in terms of taxa, the indicator species, the strategic monitoring priorities that we've already heard about. How do we choose those? And then what tools do we need to put in place to make monitoring them easier across the UK? So that's a very brief whistle-stop tour of what I'm doing at the moment. And then going forward, we've, as I've said, been experimenting with these DNA and acoustic methodologies. And now we're going to work out how we can implement them in our gardens and at partner sites to generate long-term monitoring data. We've got the ongoing sector workshops, which I'll be inviting as many people to attend as possible, and hopefully we can get a lot of people who are here today involved in that. One of the main subjects of that, as I've mentioned, will be talking around and working towards a list of what we should monitor in urban environments. Over the next 3 to 4 years, we'll be creating a series of resources and toolkits and sharing those, and also launching the Urban Nature Training Programme. The works in our gardens start this autumn, and the UNP runs until the start of 2025. So what we really want to know through all of this activity is how can we best support your and wider interests in urban biological recording and monitoring. So please do get in touch with myself or John Tweddle at the museum to discuss how you might be involved, just learn more about the project, anything like that. Okay, thank you, and I will stop sharing my screen.
Brilliant. Thank you very much, Sam. It was really interesting, such an excellent project and sorely needed. And we're a bit short on time, so I won't ask all the questions that are in the chat, if you can go and have a look at some of them. But I think what I picked up was that there is certainly a lot of interest in this, particularly perhaps from people who are working in this area in councils or local authorities. So you said to get in contact with you. I guess be good to get a feel for how widespread a sort of project you want this to be. Do you want, are the workshops open to anybody who's interested, or are you quite sort of limited in who you can engage with? And is it just London, or is it the whole country?
Yeah, so very much the Urban Nature Project is a UK-wide project, and we'd be really, really interested to hear from as many people as possible. In terms of the workshops, we're going to be having a series of different sized events, basically. There'll be some really targeted group discussions that will invite specific people to, and then some much wider, large events that everyone who has an interest would hopefully be able to come to.
Brilliant. So we should just send you our details and get on a sort of circulation list.
email me. Yeah, I already have a circulation list of people who've attended previous workshops. If you're interested in attending future ones, drop me an email and I'll let you know when things happen.
Perfect. Thank you very much. That was really good. And yeah, if you could just go and check through if there's anything else that wasn't answered in the chat, that would be brilliant. Thanks very much.